So several people I'd like to just flash the photos of, because they contributed slides to this lecture, in particular to today. That's Yanai Ofran, who is now a professor at Bailan University and Boston University. Marco Punta here, who is at the Cancer Center in London. Some slides actually come from Avinash Schlesinger, who is the professor at Mount Sinai. Kaz Roshinsky, who is now at the New York Genome Center. So with that, we get into the introduction to function. Again, I do not believe that you really need to be very, very firm in the biology, but if I throw around terms such as proteins, residues, you should understand what that means. So let's begin with the simplest question. Life begins with what molecule? Water, you could say. We could say carbon, you could, but I'm actually talking about a so-called biomolecule, something that is specific to life, that is only, in some sense, largely only life. So what I mean is RNA. So life, the idea is that life begins with RNA, because RNA replicates itself. RNA is a molecule that has the features, essentially a lot of the features of what we typically attribute to life. Now the problem with the RNA world is that it does not really have a lot of enzymatic activity. Enzymatic activity means there are certain reactions, cutting bonds, that in fact RNA alone cannot do. Okay? So what's the idea? How could RNA do that? And if you were to be living in the RNA world and RNA would have an engineer and you would be that engineer, what would you do? Or if you went to the tube and you know RNA alone is just not enough, you have to contribute something to it. So the answer is not water in this particular case. You need something that has an enzymatic activity. Any idea what that could be? Yeah, essentially a short protein or something that is the original proteins in that sense. They were small peptides, meaning just a few amino acids stuck together, and actually they do have enzymatic activity that RNA doesn't have. So in this image, we really, in fact, have a RNA enslaving proteins to do enzymatic functions for it. So the replication, in some sense the replication of information you may call, is sort of the primary master of life, if you want. The master at this point needs a slave, proteins in some sense, right? Now, if we had that image, so proteins do bind RNA, proteins do enzymatic activity, and what does it tell you about what we expect to find today in the world of proteins? Any idea? So this idea that RNA starts life and proteins are enslaved to do enzymatic function, they do RNA binding, yeah?
You have a diversity of proteins with many different functions?
You will have that. Well, actually, so now you're talking about evolution. So now you're postulating something new. The process of evolution by which you, in fact, can adopt new functions. Strictly speaking, in the beginning, in this logic, in the beginning, what we assume is that the proteins that are enslaved to do a particular job. That means there are certain things they will do right that RNA cannot do, that help RNA, that make the survival of RNA, that make the RNA world stronger, which does not necessarily mean it goes out and over time it evolves into getting new functions. That's not necessarily implied. But what is implied is that these proteins will in fact bind RNA. They will do, they are selected for doing certain things. And that's what we believe today. We believe today that the 20 amino acids that we see are not happenstance, are not chance. Those 20 amino acids just help to do that job. Life could have taken other amino acids, but this subset may have been just right for this early state. And then you're totally right, because it can adopt, it can change. Those 20 were enough. So in that sense, the evolutionary idea that you bring in was essential to make this enough in some sense, okay? Now, the other thing that we do believe today is that the original way of binding RNA So what I show here is particular protein structures, and there are people such as André Lupas in the Max Planck Institute in Tübingen who dedicate their life's work to essentially tracing back in history the original parts that bind. So he wants to see his idea or his question, and there are people, several other people who do this, but André is pushing this more than many. So if this really is true, this idea that RNA starts with proteins, then maybe, he argues, we will find some origins in today's proteins. We will see little parts in today's proteins that did this job in the olden days, at the beginning. And he's trying to trace this back. And here are examples on this slide where he argues that you can actually trace it back. Now, if you now started life with this, you have RNA, you have proteins, what else do you need? You have an ocean in which you swim. This whole thing happens in water or at the edge of water.
There's something to enclose it, like a membrane.
Now is that from last year?
Yeah.
This is the one, this is the introduction, I sort of duplicate some issues. Yes, life is a lot about self. And self means you have to define the difference between inside and outside, the difference between what is you, what is life, and what is the outside of life. And that ultimately, in order to do that, you have to wrap something around you. Now, if that something that you wrap around you would be plastic, it wouldn't work. Why? For people who haven't been in last semester's lecture. Any idea why a plastic bag? Okay, plastic doesn't exist in nature. This is not the answer I'm looking for. But something that is such as plastic, completely not able to let things through.
There's no interaction with the outside?
There is, sorry?
No interaction with the outside.
Which is good, right? I'm happy with myself.
Do you need the signals?
Signals, why do you need signals? We have a lot of lunars in the species of human who live somewhere in the way up in the north. They don't have interaction, and it's not true. But in principle, why is it so crucial for me to have an interaction? I mean, for a single cell to have an interaction with the outside. There are two aspects that are absolutely crucial of interaction. But what are those? And again, sort of in this evolutionary spirit, go out there and find new things. Cells do that, but this may be essential for evolution, but it may not be essential to survive. Again, these things are coupled, but there are two things that are absolutely essential. What do we need to absolutely exchange with the environment? As a cell, as me, I cannot go into my room for even for a week without any exchange with the environment. What do we need to do?
Well, we need some kind of energy source.
Yeah, we need something into us. We need food, we need drink, we cannot live a week without water. That's one direction. And there's another direction. We have to get it out again. So we have waste products that have to get out of the cell. So we in fact have an absolutely crucial exchange. Every cell needs to exchange material back and forth, outside and inside. So it gets food in, energy in, and waste has to go out. Those are absolutely crucial aspects of survival, not of a single cell for some short period of time. Now that means, then, that this wrap around the cell, around self, must be a wrap that is transparent in both directions. You have to let certain things through. In life, the way it's done, it's a lipid bilayer, that's the membrane, that's packed around it, and you somehow can imagine it like a medieval castle gate, and there are doors. So there are particular points where you open it. What is a little bit misleading in this castle analogy or fortress analogy is that in the fortress there are ports, there are doors at particular places, while in the membrane you can open doors, so to speak, everywhere. How densely packed, again, this is a question that is for people who have not attended last semester's lecture. How densely packed do you imagine a cell? Do you imagine a cell like a bag of water, a bag of liquid, And in this bag of liquid, things swim around. These things are proteins, DNA, and all of these kind of things. Or do you imagine it like something that is densely packed? Or do you imagine it like clockwork? Or do you like a solid state? What's your image of a cell? How densely is it packed? Anybody has an idea? So I always had this image. For me, that was a very surprising reality to find out that this image that I always had, which essentially was a plastic bag filled with water and which material swims around, so that everything that swims around there can easily find its path. That's absolutely wrong. What is right is the image of clockwork. Oops. Maybe I hit the wrong button. Or maybe just crashed. So imagine a cell like a watch. Yeah, this thing crashed. So this is full of material. It has the density of a solid state, and so some of these here are the tracks on which things. So this is in fact a drawing from David Gutzel, but his drawings are very much informed by images that we really have. And typically these drawings portray something better than any image that we have can. So cells entirely packed. Again, I talked about it briefly in the last lecture. DNA codes the information. RNA is sort of the intermediate step today. Proteins are the machinery of life. When we look at simple organisms, do you have any idea how many proteins, how many genes are in a simple organisms? In a simple organism, what is the smallest organism? How many genes do they have? Any idea? So the smallest thing, Mycoplasma genitalium, has about 500. Bacteria, Haemophilus influenza, Methanococcus here, there's an archaea bacteria, they span from something like 1,700 to 5,000 to 4,300 here, E. coli. This is typical sizes of bacteria. So again, they are creatures that live in a single cell. Now we go to the next one here, yeast, Cerevisia here, Baker's yeast, has about 6,000. Then we get to a simple worm, that's Drosophila, a fruit fly, to C. elegans, the simple worm. We get 17,000 here for the insect, 21 for the worm. A plant, Arabidopsis, 27. Rice, this number still have to look up and confirm this, is more than that. There are plants that are much larger than this. And we have about 20,000. Okay? This is roughly, the number is still not quite clear. What people have their hands on is 15,000, meaning in the lab we have now isolates of 15,000 proteins, roundabout, but the current belief is about 20,000. Now, it is this intermediate step idea, RNA is not quite right. RNA can also act alone. We briefly mentioned that before. It is not only intermediate step, it has some enzymatic activity, but we're going to ignore that for the time being. So protein function through structure. When protein function through structure, I obviously have this idea of there is a protein. So what is a protein? And it turns out that this is biology. Welcome to biology. This simple question is not that easy to define. One simple way in which I can define, I write down the amino acid sequence. So the amino acid sequences are simply all the letters strung up these amino acids, the 20 amino acids, 20 different letters. You write them like a word. Okay? This particular protein here is a potassium channel, or a potassium translocating ATPase, whatever it is, from E. coli. This is the number of letters amino acid it has. Now, you take this chain, throw it into a solvent, and typically all the proteins in the cell are within water, in a water environment. And what Epstein and Anfinsen, Christian Anfinsen mostly has shown, got a Nobel Prize for that, is that essentially for most proteins you just put them in water and they adopt a unique three-dimensional structure. Unique three-dimensional structure means that the same thing will always look the same in 3D. Okay? And that you don't need anything else. So there is no necessity to have another protein next to it. There's no necessity to have anything else other than protein and water. You take the string, put it into water, solvent, and essentially it adopts on its own its three-dimensional structure. You need a certain temperature and all of these things, that's true. And this is also not true for all proteins. We know that too by now. In particular for the ATPase that I showed you before, it's a little bit more complicated. But in principle, for many proteins, this is the way it is. And in principle, this is the truth. Is that a question or no? So different proteins have different structures. Here are just some. And in green, you have the beta strands, in red, the alpha helices. This image here, taken from Christine Orengo, just shows you a few proteins and the way they look. They look different. Now, when we get further into larger machineries, so this is a transmembrane protein here. Those are ion channels. Then you see that, in fact, you have aggregates. You have things that are larger than these small proteins that I showed you before. And they are aggregates because different proteins come together or different subunits of the same protein come together. And that ultimately, whether it's different proteins, different subunits, that also is the problem of the definition of what is a protein. Now, but ultimately it all starts with a sequence, such as this one. And in this particular case, this ATP synthase here is a protein that has a membrane region. So these rods here indicate the lipid bilayer. This green, blue, red is in fact the protein. That is part of the protein, and that is part of the protein. So it's a huge thing. And this particular one up here is, you can imagine, like two fingers that are intertwined. Or some of you may have seen in the olden days there was a drill, a very simple drill, where you had two wires intertwined, and you had something that moved down these wires, and when you moved this down, actually it turned, and you could drill a hole with it. So you move this up and down. By moving it up and down, you drilled a hole into the wood. And so this is the part that moves. This is the drill. You can imagine that the drill drills a hole into the membrane. And that thing here does exactly that. Okay? This big mushroom up there moves. Here you have these intertwined fingers, and they drill a hole into the membrane. ATPase synthase really works like that. It is activated by some sugar. Now, let's get back to the story of the protein. Typically, what we have is we have a database protein such as this one here, cytochrome P450. Now, the naming in biology very, very often doesn't make any sense. So in this particular case, P450 simply means that's protein 450, and 450 in this particular case refers to some mass, kilo or Dalton, which means that it has roughly 400 residues. It happens to be the case. So in this particular case, in cytochrome, it's the same story. But this particular name makes sense. It's sort of one that has been named a long time ago. It's one of the oldest proteins that exist. We can really see in history for at least in the order of at least 600 million years back, we can see this protein. And actually, again, this is an example where Michael Bodden in Brisbane, in fact, has sort of, in a simulation in computational biology, sort of tried to figure out what was the ancient protein. The P450 is there since 500 million years. So can I find out from all the P450 that I see today, what is the ancestral P450? And if I find that ancestral P450, can I put that into the tube? And they did it. This really folds, and this really has activity, and it's actually heat resistant. So this happens to be the thing that helps spearmint taste like spearmint. Now, up here, we have another protein, and assume that you only know the sequence. So I have not really removed, on my slide there is a mistake here. This should have been removed here. Just knowing it's a protein, knowing the sequence, comparing this sequence to that sequence, you sort of see it looks similar. And since it looks similar, this particular case comes from peppermint, since it has a similar sequence, you can guess by the comparison that in fact this is also a cytochrome P450, and in fact it's the same thing that in peppermint does what the other one does in spearmint. So you have the same protein in two different plants. For one you know what it does, and since the other one has a very, very similar sequence, you assume that that one does the same thing. Genetic code means since you start with a nucleotide alphabet of 4 different letters, you want to blow that up into an alphabet of 20 letters. In order to do that, you need to, with 2 mers, you can get 16 different amino acids. With 3 mers, 64. And that's what is called the genetic code. Since 64 is more than 20, you have some triplets here, that this is the amino acid C, and all of these 4 triplets here, the nucleotides CUU, CUC, CUA, CUG, they all do this, make the same. If those 4 come together, they all make a C. Okay, and that's the genetic code. So which triplet does which amino acids written down here? Again. Here's a way to sort of, in a poster from Sean O'Donoghue, where they try to sort of look at what is the reason for combining, or what do we learn from this combination. Anyway, how many proteins are in humans? So slightly over 20,000, and I briefly mentioned that already. Proteins are from 30 to 35,000 residues long. Here is protein length, number of proteins for some database a few years ago. And so one thing that you see in this distribution here is that quoting an average or median is not really, for this distribution, is not really appropriate. Yes, proteins on average are sort of 400 something residues long, but you see there's a wide difference, and again, there's an amazing tail here that may not quite make sense. Now, let's get back into this issue, or bring up this issue of big data in this field. When our lab moved from Columbia University to the TUM in 2009, the database had about 8 million proteins. Now, when I did this slide 4 years later here, the database had increased to 35 million. So it had roughly doubled every 2 years. This is absolutely in the green, because computers, well, at that point they did not quite double every 2 years, but close to it, right? So the biodata did not grow much faster than the computers did in the time. But the problem is, when we actually want to find out whether this is a cytochrome P450 in peppermint, when I know the one in spearmint, these numbers don't matter. What does matter is the number of comparisons I have to run. And the number of comparisons, if I do all against all here, is 64, 6.4 by the 12, right? 4 years later is 1.2 by the 15. That implies 20-fold increase. So the number of computational steps that you have to do increases 20-fold every 2 years. Now that no longer, this completely outgrows computers. So my molecular databases grow at least, or ballpark, 10 times faster per year than computers. So I'm going to be, in 2 years from now, I'm going to be, with the latest computer, I'm going to be 10 times behind what I could do 2 years ago. And that really is true. And that is really a problem in the field. And to solve or to address this problem, people try better algorithm, try more hacking, so finding shortcuts to the problem, try to reduce the problem as much as they can. Because this problem essentially exists, and as you see, this is already 4 years ago. Now, by the way, there's another aspect here, the aspect of data. When we moved with 5 terabyte. And in 2009, those 5 terabytes still fit on a hard disk that I put into my carry-on. So So this was so important that all the lab data was in a few hard disks that I had in my carry-on. It was a big argument then to get them into a carry-on. But anyway, so I could still do this. 4 years later, the 150 terabyte, you could not put into a carry-on. So the other aspect is to just write 150 terabyte in 2013, 4 years ago, was more complicated than doing the 5 terabyte 4 years before that. So yes, computers double, but not everything about it. And there are some problems that really come from numbers. Proteins do consist of units. So one unit is the domain. Most proteins have several domains. And one image here of a domain is shown, where this is a large protein, this is a smaller protein, and there is a similar region here that is in both, in the larger and the smaller one. And this is what we could typically refer to as a domain. So this is one domain, and this is the other domain. Sometimes, depending on details, this could be two domains here. So that somewhere here could be the cut. Now, sorry? So there is no strict definition, thank you for asking. There's no strict definition of a domain. The operational definition is the following, that when I cut here and I put it into solvent, it develops a unique three-dimensional structure on its own. Then I call it domain. Okay? So if I sort of isolated that region and it does it, then this clearly is a domain. That may not always be the case. So one other way here could be you say that inside of the domain you have more contacts than between the domains. So you essentially create something. Imagine this would be the case if you had two spheres that are connected. The density of the connections inside of each sphere would be much higher than the connection between the two spheres. And then you would say, okay, this is the situation for cut. I have two globular objects that both are sort of wrapped in solvent. They may not fold on their own. They may need this connection. They may, whatever the reason for that is. But at least I can recognize that these two form independent units. Okay? And that could be, so we would have to turn this around in order to really see whether that's the case. So this looks a little bit like it, but actually the helix lies such, I would have to turn this around to see whether it's the case. Most likely in this particular case it's not. But it could be. So it could be that, you know, when you turn this around, that you see that actually where this rod here lies, behind it there is a hole. And that actually there's only a few connections between the two spheres. Another way of defining a domain is that people argue, okay, if I cut this here, and I see say I could cut here, and I see this part that I cut away, maybe it's not falling on its own. Maybe this definition of they look like spheres is not quite right. But I see the same thing in very different proteins. I see the same thing. It's not 5 residues, not a motif. It's something longer, and it's repeated in different parts of life. Then I also assume it's a domain. So sorry for the long answer. I could have just said it's not that easy to define a domain. It's not that clear cut. But once you have a structure, you have a fairly good idea how to do it, and two experts will still disagree on it. Okay? But what is clear is there are units. There are most proteins have more than 2 domains. And what mediates protein function is the same sequence always folds into the same 3D structure. And the same human protein always, now same means 100% sequence identity, essentially always has the same 3D structure. Now there are exceptions. So I said that you take this chain, you put it into solvent, it adopts a unique 3-dimensional structure. That is right. But if something is in the vicinity, this may change. So if the surrounding environment is not only just water, but there is something charged, there's some DNA next to it, then it may actually be different. Or if the environment in which you fold is slightly different, in different parts, in different cells, you have slightly different pH values. And different parts of our cells, eukaryotic cells, in the nucleus, and we will get back to that issue, in the nucleus, in the cytoplasm, which is the part outside of the nucleus, so the cell membrane, and then there's an inside, that's the cytoplasm, and then inside of that, in eukaryotic cells, swim other compartments such as the nucleus. And nucleus and the cytoplasm have slightly different pH. So this could lead to a slightly different three-dimensional structure. Now, the 20 amino acids are shown here with a chemical formula, and briefly they have different properties, biophysical properties. One is the R and the K, under some conditions the H carry a positive charge. The D and the E carry a negative charge. Some are polar, here the T, Y, S, Q, N. I don't want you to take away that K is a positive charge. I will not ask that anywhere in any lecture, and it will not necessarily help you. What is important for you to remember is that there are different biophysical features of side chains, okay? And I just named them here. Don't worry, anyway they're yellow, you can hardly see it. So they're polar. Here the green ones are hydrophobic, water phobic, being enemy of or dislike. So they try to stay away from water. So essentially these green ones here try to be next to each other in a protein that folds in a three-dimensional structure. Now, structure essentially is the way a protein is arranged, and there are different ways to look at it. So this here are two different proteins coming together, a blue and a red. And the way this looks, so here every single residue, so every single amino acid is shown as one sphere, right? And here on the next slide, I'm going to sort of group them. Now I don't have spheres anymore for an amino acid. I'm grouping together what forms the same secondary structure. So these long blue ones and the long red ones here, these rods are helices. And they may not look similar to you, but over time you may get used, over the course of last semester's lecture, I just showed a lot of these images, and when you see a lot, you begin to sort of see that this could be a helix or not. Anyway, so these are two different, both of them are the structure, what we refer to as the structure, because the structure is defined as the atom position in 3D. And both have different sort of set of features in some sense. It's the same object, right? But in one representation here, in the sphere representation, you can see some aspects of it, and in another one here, you can see other aspects of it. Both are the same reality. Now again, so here's the ADH, the alcohol dehydrogenase, that everybody in this room here has, but some of your Asian friends may not, or do not have that, and that's the problem. This breaks down alcohol. So if you had too much alcohol, at some point this helps you break it down and feel better the next day. Now ADH, in this particular case here ADH5, needs to come together as the same protein. Two versions of the same protein need to come together. Otherwise it cannot function. Okay? So there is a homodimer. Homo means the same, dimer means two come together. So two of the same come together. If the homodimer is not formed because of some mutation, for instance, in the sequence, so something is done, that mutation could have happened through some random event. It could have happened, it could be a disease kind of event. This particular case, again, you can survive without it. It's not a major thing. It's sort of major after the Oktoberfest if you don't watch it. But anyway, so the function here also depends on the assembly of the dimer. Two of them come together. Now, what is protein function? And I'm going to just flash a few aspects here. This is again the ADH5, the alcohol dehydrogenase. The sequence here is a relatively short thing, is shown here. This is a publication. Again, so all of these, this is the name here. It's not only the hydrogenase. The name here is a little bit more specific. There are different aspects of the name. This is ADH is a sort of identifier. There are other identifiers. There's a three-dimensional structure, and then there's a functional description. And in this particular functional description here, from the Protopedia, so where proteins essentially have this type of functional description, here it says it is this long, so it's about 800 residues long. It's an enzyme that catalyzes a fourth step in the metabolism of fructose before glycolysis, blah, 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 blah. So you see, there's a long description of what the function is. Okay, this is a human-readable way of doing it, and this human-readable way is people try to put this into numbers, and there is, and I'll get back to that, something called the EC number, the enzyme commission number, that comes with 4 digits, where the first digit gives sort of a coarse-grained idea, is an oxyreductase. It means it cuts something, and then we get to more and more detail of the molecular detail of what it actually does. The particular reaction it catalyzes is shown here. All of this is part of the function of this thing. That the particular reaction it does, that it is an enzyme, that it has to be a homodimer is also part of its function, because if it is not a homodimer, it will not function anymore. And that was already in this description here, it's part of a pathway, and all of these here are different proteins doing different things. Now, the ADH5 is only one component of a long process. This process typically is called a pathway. Here is the largest database of these pathways, KEGG, Kyoto Encyclopedia. And now another way of classifying all of this together is the so-called gene ontology. And again, I'm going to give this same idea again in more detail, but gene ontology essentially, as before, is this EC numbers, the enzyme classification or commission numbers, tries to put function into numbers. It's an ontology, a hierarchical ontology, that tries to sort of group all of the possible functions into a hierarchy. Some things are sort of more detailed, some things are more coarse-grained, and put this into a computer. It wants to be a computable environment. It's not quite there yet, but this clearly is the best way to get there. Now, protein function, that means, has different components, different aspects. There's a sort of chemical aspect, so how a particular bond is, how a particular atom is bound. There's the biochemical, that something is a transferase, that something is an enzyme. There's the cellular example, that it's part of cell cycle, or part of a particular different process. Then there is sort of the developmental aspects, so when is it regulatory, when is it done, there's physiological related to a disease, genetic, dominant or recessive. In some sense you can argue that the function of a protein is anything that happens to or through a protein, and that combines all of these different processes. So this is one way of looking at it. Now, to predict this from sequence is quite a challenge. And the only way in which you can actually do it is by the combination of evolutionary information and machine learning. I'll explain in a minute what I mean by that. Now, this is an outdated slide. It's from 2000. This ends here 2003, and it shows the growth of databases. This is genes alone here. This is proteins with annotations, and this is proteins with known three-dimensional structure. Now, the red one here, so the blue one is proteins for which we essentially know the sequence, or genes for which we know the sequence. The red one is those for which we have done some experiments. And you see that the red one and the blue, the further this goes here, and this is already 15 years ago or 14 years ago, the red and the blue completely went apart. This story got only much worse. Now, when we ask how much of the human genome or the human proteins, again I said we have roughly 20,000, how many of those do we know function? And the answer is 10 to 50%. So why do I say 10 to 50? Why do I give such a large margin? And again, both, even 10 and 50, both are sort of wild estimates. the way, one issue here is the annotation precision. So when I say 10 to 50%, what do I mean by the function is known? Do I really want to know every single detail about the function? Then it's more on a 10%. Or do I care less? So if I call this whole thing here blue, then it's 50%. If it's 10%. If I call this whole thing here blue, then it's 50%. So if anything is known, then it's also a matter of how accurate or how detailed for you function should be. So if you call that an aeroplane, then sort of 10% is known. If you are happy to call this one here an airplane, then sort of 50% is known. And this is essentially what I mean, that, you know, roughly having some idea that something looks like an alkyl dihydrogenase, I call that knowing function. Then essentially we have 50%. That's the uncertainty in the numbers. Again, what we know is we take a protein, we throw it into solvent, it develops a unique three-dimensional structure on its own. In principle, let's just revisit the question, how well can we predict this? In physics, typically you have situations described, we are in the right room for that. Physicists describe things in terms of energy landscapes, and in an energy landscape you can predict that if the ball at some time point here is starting at this position and you wait a little bit, then you can absolutely predict where the ball will be. You can even do the prediction accurately if there were a little bit of a mistake in the initial conditions. You still would predict the outcome of this experiment very accurately. In contrast, if you had this situation, then you would immediately see that tiny mistakes in the initial conditions here would make a mistake in the way you predict. And sorry for the animation, something went wrong here. Now, that means that essentially you have a situation in which you need to know every single detail about a protein. Or putting differently, point mutation changes function, binding changes function, the environmental change, so something next to it could be DNA, yes or no, that will change the function of that protein. And if that is the case, then ultimately you need to know the history of the molecule, not protein ADH5. You need to know the history of this particular ADH5 in this particular cell at this particular time point in order to predict it. You need to know what is next to it, right? And that sounds exactly like this type of problem. That sounds like what physicists call chaos. That's the type of problem that you cannot solve. It's the butterfly effect. That's one way in which is sort of commonly known. Tiny differences in the initial conditions will ultimately completely change everything. So, and here is the reason why it's not that hopeless, because in fact evolution has exactly this history. What I show here is number of residues aligned, percentage sequence identity. In the blue region are protein pairs that essentially have similar 3D structure. Now let me pick an example here. If I have two proteins aligned over 100 residues and 33% of those residues are identical, then essentially they are in the blue region here, and then essentially they have a similar 3D structure. That means you can change 67 residues and not change structure. Okay? Now comes the surprise. I go to the lab, I change 5 residues, and most likely, randomly, right? Or no, I pick some 5 and I change them. Most likely, I'm not going to maintain a structure. How come? Nature can change 67 and I cannot do 5. So one valid answer is because I'm an idiot or I'm not very good at it. Nature is much better. Fair enough, I take that point. But there's a little bit more to the answer. Anybody? Yeah?
There are certain amino acids that are crucial to so that the function is still maintained if you...
How come you're totally right? And how come nature knows them and I don't? Again, I gave you an explanation that is part of the reason. This is true.
It's part of the evolution, so to figure out that these amino acids, they are really important to...
That's exactly the point. So evolution is in that corner. There's two evolutionary arguments from the two of you. So ultimately, a neutral change is very unlikely. Most likely, most changes will have some effect. But only the neutral ones will be observed. Only the ones that do not change the fitness of this thing to do the right thing. Those you will see. Or sometimes you will see things that will be selected for that do something better or that do something else. Those things happen. But ultimately, things that will not work will all not be seen. In this comparison here, I'm not doing any invented protein or any in silico possible protein by any in silico possible proteins. I only compare proteins that are known, and they are selected for. Okay, for exactly that mechanism. So evolution had enough time to make an experiment of the very unlikely event, which is neutral variation. Now, this implies that if I now started with a family of proteins, so I take a protein, I find proteins that are very similar in terms of their sequence, and I build up many in the database. So I have a family of related proteins. Of all of these, for instance, they're all P450s, or all ADH5. So I know they have a similar function. I know essentially they have a similar structure. But somewhere, at some places, they will change. There's 67 and 100 residues. They will appear somewhere, right? And I know that they are neutral. Now, I'm going to show you on the next slide a particular family, SH3. And in this family, is a very old slide. In this family, there are 4 essentially conserved columns. 1, 2, 3, 4 in purple here. And these are 2 different sites of the same molecule. And you see that the first one here and the last one, in 3D, they are close to each other on the surface. These two, oops, yeah, I'm sorry. These 2 residues are together on the same side of the protein. Imagine, so something, what I wanted to say, they are far away in sequence, so from here to here, close in 3-dimensional space. Now, since evolution acts on the 3-dimensional object, anything that happens here has an impact on what you see here. Just imagine that that replaces, say this has to be a salt bridge, meaning a positive and a negative residue. And imagine that this suddenly here, the positive residue, is flipped to a negative one. In order to maintain the bond, all you need to do is flip that one to a positive, right? The other way around. So you maintain the bond, right? But the maintenance of the bond would be something that is correlated between this position here and that position. Put differently, assume you put 5 consecutive residues out of this profile here of all of these observations of amino acids, so 1, 2, 3, 4, 5 in this window here. In these 5, and what you see in this change there is some indication of what happened here. This means that if you see these 5, or the change window in these 5, you can see some event that happened somewhere else. It's like in the raindrop in which you see the world. Okay, you see a reflection of something else. You may not be able to interpret it right. You may not be able to really see through, okay, that positive residue, and you don't know where it was. It could have been a positive residue here or here. Who knows? But if you see the same effect, for instance, in this example here that I gave, say all of these are positive and then suddenly they switch back to negative, and here you have the opposite, then you know exactly which ones are correlated. You know exactly where it came from, right? So you do see not only the reflection, you actually see the truth. In terms of the problem what we have here, this helps you to somehow convert the chaos problem into a solvable problem by essentially averaging over different species. Now, averaging over different species was done first historically in the sense by feeding a profile. A profile is essentially, you look at a family of proteins and you count at every single position how many A's are at that position in that family. And that whole thing you call the profile. So essentially the observation what you have. And you put this profile into a machine learning device, and that machine learning device can predict secondary structure. This is one example. There are many other problems that have been done with this same idea, and several of these we will talk over the course of this lecture. For instance, the prediction of protein-protein interactions. If we succeeded, we could do many things with this. We would have medical applications, we would reduce the cost of molecular biology, and many, many other advantages. So there's a big advantage to be gained if we push the envelope. Let me repeat, and this is a slide from Luisa Arura. This is again one particular protein, leptin. And in this particular protein, sorry, the animation, this particular protein has in different cell types very different roles. It's the same three-dimensional object, and I did not put I forgot to put the three-dimensional structure in here. But depending slightly on the environment, it can have different effect as the readout. So this one is related to some disease. That is what Anna here worked on. This particular one here has to do with anorexia or obesity. It's a very complex mechanism how something that is related to anorexia leads to obesity. Anyway, so the major point here is you put the same molecule into different environments and it does slightly different things. Molecular dynamics means that essentially function is motion, and there are different time scales of motion. Let me just hope, yeah, it does work. So this is one protein that the type of thing that I showed you before. And as you see here, the secondary structure segments, purple helices, yellow the beta strands, highlighted. So the crowded, high density, noisy, essentially there's a lot of movement of the protein. They're very dynamical objects. And some of this movement is shown here and is realistic, so you may somehow hear the sound that I took off. I take off even more. But the idea really is of this movie to show that in fact function happens through movement, and proteins are very dynamic beings that move a lot. And so briefly, DE show, and I'm not going to show this. David is not visible here. So here are some of these movements, and you will see that ultimately, I hope the motion is long enough, that these are pushed through these molecules here. But that is typically the type of movement. David Shaw built a giant supercomputer, Anton, that is optimized to simulate protein folding. And with that here, he shows how a particular drug finds the binding site. I'm going to skip through this. This is just one YouTube talk from David Shaw. It's an outstanding piece of work, and there's nobody in the world actually as far as they are. Now, so far I've always talked about a particular protein chain adopts a particular three-dimensional structure, and that is relevant for function. Now, that's the world of order. There's something else that is disorder in the world of proteins, and this is not a disease, but it is something where this protein chain here is not adopting a unique three-dimensional structure. It's essentially dangling around as it is. Okay? Unique three-dimensional structure I would define as something that I shine light at different time points and I would see the same thing. In this particular case, I would not see the same thing. Okay? Now, it meets with a protein, well-folded protein, and upon binding to that protein, it adopts a unique three-dimensional structure. So now I shine light on this at two different time points, and the red will be the same. Okay? So upon folding, it adopts a unique three-dimensional structure. But back to the issue of the question of domain, this is a protein. It just does not adopt a unique three-dimensional structure. And it seems that this type is referred to as protein disorder. Some of the protein disorder is such that it adopts a unique three-dimensional structure upon binding, others doesn't. And there are many reasons why it may not adopt a unique three-dimensional structure. One essentially is this image of filling material. So when you buy something, some electronic equipment, you have these bubbles, these plastic bubbles, in order to avoid that there's some damage to your electronic equipment. And this is the same story here. By not adopting a unique three-dimensional structure, you move around, you keep big space. It is actually like a bubble, okay? And it seems that, in fact, eukaryotes adopted more disorder, and through more disorder they can achieve more complexity, or some operations of higher complexity. Okay, now I already said protein function is intuitive but not well defined. Let's just look for at a couple of metrics for protein function. One is very simple. You can define, or you can compare sequences. You can compare sequences by counting how many letters are identical, how surprising a similarity between two sequences is, an e-value. The same is true for structure. It's very simple to measure similarity in terms of the atomic positions. You could imagine that you look at corresponding atomic positions and you define a distance between them. Root mean square differences here. Is the RMSD. There are different ways of doing it. Now, what about function? And one way that I already introduced here briefly is the enzyme nomenclature. In the enzyme nomenclature, where on the first digit you have 6 different numbers here, the oxyreductase is what I showed you before, transferase, hydrolase, lyases, isomerases, and ligases. Again, this is only for enzymes. And then we get further down. So if you are lyase, they bring 2 different atoms together. And now the question is, are these 2 carbon and carbon, or carbon oxygen, carbon nitrogen, blah, 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 blah. So the next digit essentially is, what are the 2 that you put together in this particular case? And then you get into further detail. And this way you build up a number with 4 digits that is completely hierarchical. In the gene ontology that I presented, we have a similar idea. Now, on the first point, I said there is something that is function that is sort of the molecular, the way you bind, the enzymatic activity, how it's done. And then there is something on the level of pathway. So which process are you part of? And then there is something about where in the cell do you do this. And that is, in fact, why Go, the gene ontology, begins with 3 different hierarchies. So it has a hierarchy for each of those. It says, well, there's molecular function. I have built a hierarchy for molecular function. There is processes. I built a hierarchy of processes. And then there is sort of places in the cell where you can be, locations. And those are 3 different hierarchies. And then the protein is every protein is part of one of each of these 3, right? And here is a particular example for a protein in the pathway process, where at some point here you have DNA priming. So the whole thing is DNA metabolism, so it has to do with DNA. And then there are further details what you actually do with DNA. Then there's molecular function. Again, in this particular case, on the highest image here, you would say it's nucleoside binding. Then it's actually DNA, not RNA binding. And then more detail of that. And the cellular component, you would say the binding here is in the nucleus, and a different particular part of the nucleus is where it happens. Now, again, those are examples that I showed. Now, it is not a strict hierarchy, meaning that, so one aspect of it not being a strict hierarchy, you cannot always cut at level 1, 2, 3 and say everything on level 3 is comparable in terms of level of detail. That depends exactly where on your tree you are, where on your path, where on the ontology you are. In different DNA binding may have a higher depth the tree for DNA binding may have a higher depth than the one for alcohol binding or something like that. Now, I'm going to zip through these here. The domain problem I already, and let's begin with a recap. I do like to repeat this. I briefly mentioned it in the last lecture. So often I will show examples where I show one protein running from residue number 1 to residue 511. Another protein here, which could be from a different species or from the same species, running from 1 to 327. And this overlap here means that there is something aligned, meaning that there is some significant similarity between these two proteins at that point where they overlap. So in this particular case here, we have 3 proteins that have a region in which they overlap. And again, to the definition of domain, I gave you 3 examples how we can define domain. None of those is shown here. What I show here is that when I see that this protein has that region in common, that protein essentially has the entire region, then I may argue this may be, even if I don't know the 3-dimensional structure, if I have not put it into the tube, I don't know anything about it, but still, maybe this is what could be a domain. Because here's a protein that I do know exists alone, so that without even doing the experiment, nature has done that experiment for me as soon as I find that protein. So there is something of that length that folds on its own. And maybe then, since these could be 2 different domains, maybe that then would be the domain. But again, you see there's a lot of leeway where exactly you put the boundaries, where exactly you put the cut. So, well, here you cannot cut, but you could cut a little bit further here. But here you cannot cut, so maybe the domain is essentially shorter than that. Difficult to say, so there's leeway. Let me recap. The first step in the introduction to function, the first step, it's the second level of batteries. The first step, you have to find related proteins. Those are alignment methods. We discussed in the last semester a lot. We will not really need to know how they work in this semester, but that something such as alignment methods exist is important. They produce alignments, so comparisons of entire families. To know that is important. How exactly that is done is not impossible. It is not important. Over the course of this lecture, we will use the output of those methods. Okay. Then. Typically, methods that do this, most prominent today is something called PSI-BLAST, because it does it very fast. And BLAST, that is able to, simply looking at the sequence, find that 2 things are related. Function is known for about 3,000 to 12,000 human proteins, so for 10 to 25%. And the other way around, again, no matter how you look at it, there's a lot that remains unknown. The goal of protein prediction is to predict from the sequence the 3-dimensional structure and actually the function. David Shaw, I briefly zipped over a talk from David Shaw. David Shaw built this supercomputer, Anton, to essentially predict protein 3D structure from sequence. And we are still at this point not quite there. So Anton can do that for short fragments of proteins. For very tiny proteins, it can actually do that now. But for most proteins that we carry in us, we cannot do it at the moment. Most predictions of 3D structure today are using known experimental structures that are somehow sequence similar. Like in this example of peppermint and spearmint, the P450, where we have similar sequences, and that's the same idea that is the most successful technique to predict 3-dimensional structure. So for one protein, you know experimentally the structure, the 3D coordinates, and another one has a similar sequence. You simply look up the coordinates of the one that you know. It's referred to as comparative modeling. That's the most successful technology to predict 3D structure. Doing this de novo approach, where I take a sequence and I don't look up any known structure, I just fold it on its own like the protein does it. Again, in some situations we can do that, but it's the rare exception. Can we predict function? Ultimately, what we'd like to do in order to predict function is we like to know that the ADH is an enzyme, that this enzyme is involved in a particular process. Ideally, I'd like to know that it has to come together as a dimer, and I'd like to know what it actually binds and where this binding happens. Those are the aspects that are minimally, I hope, to predict from sequence. If I can look up another protein that has a very similar sequence in the P450 example, then I actually want to really know the Go classification of something that I already have classified. And ideally, I would be able to look up, write up this, with a computer, write this abstract here by simple sequence comparison. But that I can only do if I can sort of look up of a related sequence the same abstract. If I want to predict, I can only predict some aspects. I can predict that it's an enzyme. I can possibly predict the pathway in which is involved. Some of these things here I can look up, and that is the state of the art. And I'm going to present a couple of methods that sort of fix some of these issues. To accomplish this, essentially the way the field has been trying to do is to develop one method after another that does a particular step. One step is, for instance, where in the protein, where in the cell is the protein native. So that's the localization. And we're going to talk about localization prediction. Another, every protein interacts, so almost no protein acts in isolation. All proteins, so they touch. I said it's the cell is packed as densely as a clockwork, so everything touches everything, but everything also actively interacts with something else. And so predicting the binding partner is, I'm going to present methods that predict protein-protein interaction, that predict protein DNA and protein RNA interactions. In fact, I'm going to zip over a few methods that predict binding to small substrates. And